Welcome to Radiology Essentials, Cells, Molecules, and Radiosensitivity. This video will explore the fundamental building blocks of life and how they interact with radiation. In this overview, we'll dive into human biology fundamentals and the critical concept of radiosensitivity, vital for understanding radiology. Let's start with the cell theory, the cornerstone of modern biology. Cell theory explains that all living organisms are composed of cells, the basic structural and functional units of life. The key principles are: all living organisms are made of cells. Cells are the basic unit of life, and all cells arise from pre-existing cells. Next, we'll explore the molecular composition of the human body. The human body is composed of essential molecules like water, proteins, carbohydrates, lipids, and nucleic acids. These molecules play vital roles in cellular function, providing structure, energy, and genetic information. Let's now delve into the intricate parts and functions of human cells. Cells contain major organelles such as the nucleus, mitochondria, and ribosomes, each with unique purposes essential for life. These organelles work together in a coordinated manner, enabling cells to perform all life processes, from energy production to waste removal. Understanding cell division is crucial. We'll briefly discuss mitosis and meiosis. Mitosis involves a single cell dividing into two identical daughter cells, crucial for growth and repair, occurring in distinct stages. Meiosis, on the other hand, is a specialized division that produces four unique cells, essential for sexual reproduction and genetic diversity. Finally, let's explore radiosensitivity of tissues and organs, a critical aspect in radiology. Radiosensitivity refers to the susceptibility of cells, tissues, and organs to damage from ionizing radiation. Several factors influence tissue response, including cell division rate, differentiation, and oxygenation, all important for predicting radiation effects. Highly sensitive tissues include bone marrow and reproductive organs, while muscle and nerve tissues tend to be more resistant. In conclusion, understanding cell theory, molecular composition, cell functions, division, and radiosensitivity provides essential insights for radiology practices and patient safety. Welcome to our deep dive into the fundamental principles of radiobiology. Today, we'll explore how radiation interacts with biological systems. Radiobiology is a fascinating field that examines the effects of ionizing radiation on living organisms, from molecules to complex systems. We'll cover several key principles that are crucial for understanding radiation's impact. First, let's look at the law of Bergoni and Tribando, a cornerstone of radiobiology. This law states that the radiosensitivity of cells is directly proportional to their reproductive activity and inversely proportional to their degree of differentiation. This means that rapidly dividing cells, like those in the bone marrow or gonads, are much more sensitive to radiation than mature, differentiated cells. Now let's explore the physical factors that influence how biological systems respond to radiation. The total radiation dose and the rate at which it's delivered significantly impact the biological outcome. The type of radiation, whether it's alpha, beta, gamma, or X-rays, and its energy level, all play a role in biological damage. The presence of oxygen can also enhance the damaging effects of radiation, a phenomenon known as the oxygen effect. Beyond physical factors, several biological elements determine a cell's radiosensitivity. A cell's sensitivity to radiation varies depending on its phase in the cell cycle, with some phases being more vulnerable than others. Cells with a high proliferation rate and less efficient repair mechanisms tend to be more radiosensitive. Understanding dose-response relationships is critical for predicting radiation effects. Linear dose-response relationships suggest that the effect is directly proportional to the radiation dose, even at very low levels. Nonlinear relationships, on the other hand, indicate that the effect might be less pronounced at low doses, but becomes significantly more severe at higher doses, often with a threshold. Let's categorize different types of radiation and understand their linear energy transfer, or LET value. We'll differentiate between alpha, beta, gamma, X-rays, and neutrons, highlighting their unique properties and how they interact with matter. Linear energy transfer, or LET, quantifies the amount of energy deposited by radiation per unit path length, influencing its biological effectiveness. To summarize, understanding these fundamental principles of radiobiology is essential for predicting and mitigating the effects of radiation exposure in various applications.